Welcome to Produce Talks.
This whole business is based on relationship and friendships.
Food is darn simple.
What can we do? What are other countries doing?
How important it is for industry to be talking about this.
We're going back to the farm. We're going back to the whole food.
Hi, I'm Wally Burns, the Education and Training Manager at CPMA. CPMA would like to acknowledge Nature Fresh Farms as our Produce Talks series sponsor. Find out more about Nature Fresh Farms and how they are growing for a kinder future at naturefresh.ca. You can keep up with them on Instagram or Twitter at nature_fresh and on Pinterest at naturefresh, along with other social media. In today's episode, Jeff Hall, who is CPMA's food safety specialist, talks to Dr. Lawrence Goodridge and Dr. Jeffrey Farber, both from the University of Guelph, about whole genome sequencing. In the second part, Jeff discusses this technology in regards to food safety with Afreen Malik, Director of Science Programs at Western Growers. And now, here's Jeff.
Thank you, Wally. Whole genome sequencing has quickly become an important tool in the investigation of foodborne illness and is quickly being adopted by industry as part of their food safety programs. There still remains, however, a knowledge gap on what the technology actually is, how it works, and why it is so powerful. To help answer these questions, CPMA is excited to have with us experts representing both academia and industry. Up first, we will be talking with Dr. Lawrence Goodridge and Dr. Jeffrey Farber. Dr. Goodridge is the Lung Family Professor of Food Safety, Director of the Canadian Research Institute for Food Safety at the Department of Food Science for Guelph University. After completing a BSc honors degree in microbiology at the University of Guelph, Lawrence completed his MSc and PhD degrees in food science with an emphasis on microbial detection of foodborne pathogens, also at Guelph University. Before leaving for a postdoctoral fellowship to continue his training in microbial food safety in the laboratory of Dr. Michael Doyle in the Center for Food Safety at the University of Georgia. Prior to returning to Guelph, Lawrence has held faculty positions at the University of Wyoming and Colorado State University. More recently, he was the Ian and Jane Monroe Chair in Food Safety in the Food Science Department at McGill University. In January 2019, Lawrence joined the Department of Food Science as Director of the Canadian Research Institute for Food Safety, where he holds the Lung Family Professorship in Food Safety. Dr. Goodridge conducts research related to control and detection of pathogenic microorganisms, including bacteria, viruses, and parasites. Dr. Goodridge has published more than 100 peer-reviewed journal articles and book chapters, and has been awarded more than $30 million in research funding from U.S., Canadian, and international funding sources, including CPMA's own Canadian Food Safety Fund. Dr. Jeffrey Farber is currently the director of an international consulting firm, which does food safety consulting with various organizations and countries. In addition, he is a senior advisor for Index Biosystems, a Canadian biotechnology company working in the area of food traceability and authenticity. Dr. Farber is also currently an adjunct professor and member of the graduate faculty in the Department of Food Science at the University of Guelph here in Ontario. Dr. Farber's recent employment included full professor in the Department of Food Sciences at the University of Guelph, Director of the Canadian Research Institute for Food Safety, head of the master's program in food safety and quality assurance. His public service credentials include Director of the Bureau of Microbial Hazards in the Food Directorate of Health Canada. Dr. Farber has over 180 publications and was the Associate Editor of the International Journal of Food Microbiology. He is a past president of the International Association of Food Protection and Executive Director of the International Commission on Microbiological Specifications for Food, and has extensive experience working at the international level with organizations such as the FAO, the WHO, and Codex Alimentarius. In addition to numerous personal and team awards, Dr. Farber has a special claim to fame by having a new species of listeria named after him, Listeria farberi. Welcome, Dr. Goodridge and Dr. Farber, and thank you for joining us. Now, let's dive into the questions. Dr. Goodridge, whole genome sequencing seems to be occupying a more important role in foodborne outbreak investigations, but many people don't understand what it is or its history. Can you give us some background on the history and evolution of whole genome sequencing?
So thank you, Jeff, for inviting me today to answer these questions. And I'll first begin to answer this question by explaining what whole genome sequencing is. So the genome or genetic material of an organism, whether that's a bacteria, a virus, a potato, an animal, a human, for example, is made up of DNA. And each organism has a unique DNA sequence, which is composed of bases. And these bases are designated as letters, A, T, C, and G. So all the DNA sequences in any organism is comprised of these bases, these four bases, A, T, C, and G. If you know the sequence of the bases in an organism, then you have identified its unique DNA fingerprint or pattern or sequence. So determining the order of bases is called sequencing. And whole genome sequencing is a laboratory procedure that determines the order of the bases in the genome of an organism in a single process. And so the history of DNA sequencing can be traced back to 1977, when the first sequencing method called Sanger sequencing was invented. And this actually led to the Nobel Prize in being awarded for this in 1980. And between 1980 and 1998, there were some advances made, primarily in the infrastructure, the equipment, making it easier to use, making it automated, for example. In 2000, the human genome was sequenced, and this was quite a milestone. It had taken a number of years, over 10 years to sequence, and the cost was astronomical to do that. So between the years 2000 until now, we've really seen a modernization of whole genome sequencing, primarily in the way that the sequences are determined. So that means the equipment, and also how we analyze those sequences. So, for example, we've seen what is called next generation sequencing systems be developed. And the critical difference between Sanger sequencing, that first method, and next generation sequencing, or NGS for short, is the volume of DNA that can be sequenced. So the Sanger method was very slow and only sequenced a single DNA fragment at a time. Next generation sequencing is what is termed as massively parallel, which means that this approach sequences millions of DNA fragments simultaneously in a single run. And so this means that we can sequence hundreds to thousands of genes at one time. And it also allows us greater discovery power to detect novel or rare variants, which can be used for specific applications with food safety. So where we currently are in 2022 is we have several options for next generation sequencing. And the approach has really been streamlined and standardized. And we also have standardized approaches to analyze the DNA sequences, because that's a major aspect of DNA sequencing, which we will discuss in later questions.
Thank you for that, and it's certainly interesting to hear about the evolution of the technique, and the fact that it is now something that is much quicker and analyzes much more information is certainly a leap forward, I would assume. If I could go over to Dr. Farber for a moment. At a number of conferences over the years, I've heard the term PulseNet. Can you explain to us, to the audience, what PulseNet is and how does it factor into the whole genome sequencing equation, and why is it important?
Thank you, Jeff. And thank you very much for inviting me here today. It's certainly my pleasure. So PulseNet, I can briefly describe PulseNet Canada. Each country has their own PulseNet. So there's a PulseNet USA. There's actually also a PulseNet International. PulseNet Canada is the national molecular subtyping network for foodborne disease surveillance. Now, this national network is actually administered by the National Microbiology Lab in Winnipeg, and they are actually responsible for the data collection and analysis for all the isolates that come in to their lab. The members of the PulseNet Canada are, besides Public Health Agency Canada or FAC, are the Canadian Food Inspection Agency, Health Canada, and the provinces and territories. Most of the labs now in the provinces have been trained actually by Public Health Agency of Canada. They have their own instrumentation. They do some of the initial data analysis, and then they send that data on to Winnipeg to do sort of a centralized analysis. Now, before PulseNet, often foodborne outbreaks went actually undetected or were discovered only after they grew very large. Now, what happened is that the initial start of PulseNet actually started in the United States by Dr. Swami Nathan at the CDC. And that was basically in around 1996. Canada started a little later, about 2001, 2002. And when PulseNet was first on board, they used a different type of technique from whole genome sequencing. They actually used what's referred to as pulse field gel electrophoresis, where what you do is you actually have enzymes that cut the DNA into fragments, and then you run that DNA on a gel and you look at differences in size and banding patterns between different isolates. But as whole genome sequencing became faster and easier to do, it has been seen definitely as the gold standard now and much more sensitive than pulse field gel electrophoresis. And actually, the Public Health Agency, since 2017, has now switched to whole genome sequencing for all the analysis, let's say all the bacteria that come from foods, from clinical specimens. Those are all sequenced and sent into the database in Winnipeg. What Winnipeg would do then is they keep a track of all the isolates. So, for example, if we're looking at Salmonella enteritidis, and just as an aside, Salmonella enteritidis is one of those organisms that's very closely genetically related. And we always had a very tough time distinguishing between different strains of Salmonella enteritidis. Now with whole genome sequencing, it is so much easier. The light has come on in terms of being able to differentiate between strains, even in a closely related species, such as Salmonella enteritidis. And I think for the Public Health Agency of Canada, one of the first sort of very interesting uses of whole genome sequencing during a foodborne outbreak, some of folks may remember there was actually a listeriosis outbreak due to caramel apple. And they had a lot of trouble in the initial phases of the outbreak, although most of the cases occurred in the United States. There was only, I believe, one in Canada, but still Canada did try to participate in trying to solve that outbreak. They initially were using only pulse field gel electrophoresis, and they were actually coming up with hot dogs as a source. Then when they started using whole genome sequencing to get that finer, better discrimination, they were able to actually hone it down to these caramel apples. And so I think everyone has realized really how great whole genome sequencing has been and how much of a game changer it has been in terms of solving outbreaks. So again, in terms of the isolates, The isolates going into Winnipeg, they could be food isolates that come from surveillance programs. They could be environmental isolates that are taken during environmental swabbing of a different plant. Then you have all the clinical isolates that come in from looking at a small number of cases, what we refer to as cluster of cases. And so what Winnipeg does then is they compare the whole genome sequences of the clinical isolate to the whole genome sequence of the food isolate to see if there's actually a match between the two and to see if they can make a connection. And as I mentioned before, the use of whole genome sequencing has allowed us to stop the outbreaks in their track, so not let them get very big, because we're able to solve the actual food source. Also would lead to probably less recalls because you're much more sure of what the food source is in that outbreak, and you're not implicating, either a food source that is actually non-evolved, or you're implicating maybe 2 or 3 items instead of one single item. So in summary, I think whole genome sequencing in terms of PulseNet has really been a game changer and will continue to do so in the future.
Thanks for that. And I'm just kind of reflecting back on what you'd said about the caramel apples. I remember that was certainly a weird one when it popped up. And but it is good that with the new technologies that we were able to identify the exact food source. I mean, as someone from the fresh fruit and vegetable industry, I would have preferred it was hot dogs. but you know what? I think the key for everyone is we want to make sure that we're as right as we can be, and that's based on solid science. Just if I can build that upon that a little bit. My understanding is that there's several methods that public health can use when it comes to whole genome sequencing with respect to the data for foodborne illnesses. Can you kind of briefly describe the different methods they will use?
Yes, thanks, Jeff, for that question. So Dr. Goodridge had talked a little bit about the nucleotides and the bases and Sanger sequencing. So there are actually 2 main methods. I don't want to go into great detail here, but basically there are 2 methods that are used. And basically one of them looks at comparison of the genes. So we call it a gene by gene approach. So how different are the genes of 2 different strains, for example, of Listeria? And I will just briefly describe this and then go on to the next technique. What happened was a little while back, there was a scheme which was developed, which was called multi-locus sequence typing. It's called an MLST scheme. And what happens in this particular scheme is that investigators chose 7 different genes that were referred to as housekeeping genes. So these are like genes that work in the background to keep bacteria alive and let them function and do whatever they have to do. And so what they did is they actually were able to amplify these 7 different housekeeping genes by using PCR, which many of you are familiar with now because of COVID. And then they sequenced the amplification of these housekeeping genes. And so what you can do then is you can actually compare the specific DNA sequencing of these 7 housekeeping genes from one strain to another. And if you only see, let's say, a single nucleotide difference, that signifies that as a different strain. And we actually refer to that as a different sequence type. So you may see in the literature, could be Listeria sequence type 30 or sequence type 70. At that point, that means that there was a difference in one of those 7 housekeeping genes between the different strains of Listeria. Now, because of whole genome sequencing, we've been able to actually make that technique even better. So instead of only looking at 7 genes, we are now, because of whole genome sequencing, this has been upscaled. So now we can look at hundreds or thousands of genes. So you can imagine the the great power when you're able to look at and analyze hundreds or thousands of genes and look at the nucleotide sequence of those and see if there are any difference between the strains. We refer to that, and I'll just briefly go into it. One is referred to as core genome MLST. So you're looking at what the basic core genes of an organism, and this can be quite substantial. And so then again, you're looking at a gene by gene approach of all these hundreds or thousands of genes. And we also have a whole genome technique, so a whole genome MLST, and that's using not only some of those core genome, but other, what we refer to as accessory genes. So that has really revolutionized the way we use whole genome sequencing to distinguish between strains. So that's one technique, and we refer to it again as either core genome or whole genome MLST. And that's, for example, the Public Health Agency of Canada uses the whole genome MLST technique right now. There is another technique that we refer to, and that's called SNP analysis. And I think some of you may have heard of this. It's referred to as a single nucleotide polymorphism-based approach. And here, instead of looking at genes, you're actually looking at differences actually in the nucleotides. So again, this is another technique. There have been a number of studies that show that this SNP, SNP-based approach is comparable to the core genome or whole genome MLST approach, fairly similar. In some cases, it may give you some extra discriminatory power, but in general, most regulatory agencies around the world are now using either the core genome or whole genome MLST approach. So these are the two basic approaches that are now being used.
Well, thank you for that, Dr. Farber. I'm going to move back to Dr. Goodridge now. And Dr. Goodridge, as our laboratory practices expert, can you walk us through the procedure which is followed when a sample arrives in the lab, right through to the final analytical results?
Yes. So the first thing that has to happen with respect to whole genome sequencing is this has to be done on pure bacterial isolates. So to begin, that sample will come into the lab and we would isolate the bacteria by doing dilutions and plating on solid microbiological agar to obtain separated bacterial colonies, where each bacterial colony originated from a single bacterial cell. So then what we do is we would take a bacterial colony and we could grow that up in broth so that we increase the numbers of the cells. And then we would treat that culture with a chemical that would break open the bacterial cells, releasing the DNA. And then we can use various kits to purify the DNA. Next, we have to break the DNA up into smaller pieces so that the sequencer can appropriately sequence the bases. So the DNA is cut into short fragments, and typically this would be of known length, because depending on the type of sequencing machine used, the different sequencers will sequence different lengths of DNA fragments. So we have to cut the DNA into fragments, shorter fragments of known length, and this can be done by using enzymes or molecular scissors that precisely cut the DNA into fragments at known positions. And then once we've done that, we have to make many copies of each DNA fragment. So Dr. Farber briefly mentioned PCR, which, you know, as he said, we're all familiar with the approach used to test people to see if they have COVID-19 during this pandemic. So the same type of approach would be used to amplify the DNA fragments, these smaller DNA fragments, and we make many copies of them. And so once we do that, that's known as a DNA library. And this DNA library is then loaded onto the whole genome sequencing machine. And this machine will then determine the combination of the bases that I talked about before, the A, T, C, and G, making up each individual fragment of DNA. And so once a fragment of DNA is sequenced, it's called a read, a DNA read. And we talked about earlier about the fact that these next generation sequencers can sequence many of these reads simultaneously. So this is why it's called massively parallel sequencing as well. So once the sequencing is done, the sequencer produces millions of DNA reads, and now we have to reconstruct that into the whole genome of the bacteria. So that's why we call it whole genome sequencing. So there's no way that we can do that manually. We need to have specialized computer programs that do that. And so they put these DNA reads back together in the correct order, like pieces of a jigsaw puzzle. And so when completed, the genome sequence, which contains millions of bases, is in one piece, or in some cases a few large pieces, is then ready for further analysis. And where this analysis is what Dr. Farber previously mentioned. So these would be the analysis that one could use to determine if, for example, a colony that came, or a bacteria that came from a food isolate, and a bacteria, the same bacteria that came from someone who was sick, has the same genetic footprint.
That's very, very interesting. I always kind of wondered how, after the genome was taken apart, into the smaller pieces to be analyzed, how that gets put back together again. Kind of the Humpty Dumpty scenario, but with a genome. A number of CPMA members, are looking at whole genome sequencing and wondering what role it would play within their food safety testing regimes. What advice would you have, Dr. Goodridge, for them when it comes to incorporating that into their food safety practices? And secondly, are there any certain technical or procedural or even equipment requirements that they need to be aware of?
Great. So to answer the first question, what advice I would give them. So, whole genome sequencing has really become mainstream, I would say, over the last 10 years or so. And I know a lot of members of the food industry and the fresh produce industry have really been asking, how can we employ this technology? One of the first things to understand is that whole genome sequencing produces an enormous amount of data. And so when trying to decide whether or not to incorporate this, the first question should be, what questions are we trying to answer? And what are we going to use this data for? So for example, if the fresh produce company or grower, et cetera, is only interested in determining whether or not a potential foodborne pathogen is present in a sample, then whole genome sequencing would not be recommended, because it produces much data for that purpose. And we know that there's rapid tests that could give that answer in a much more appropriate and cost-effective way. If, however, the fresh produce grower or company is interested to know more about the ecology of microorganisms, for example, in their processing plant. So let's say that there has been a persistent contamination with a given foodborne pathogen, like let's say Listeria monocytogenes, for example, in the plant, and it's showing up sporadically in their product. Then whole genome sequencing would be appropriate, because it could be used to do environmental sampling, as Dr. Farber has said, where one could take swabs from throughout the processing plant environment, grow them up to see if there's Listeria monocytogenes present. And if there is, one could then apply whole genome sequencing on the different samples to see if the bacteria are genetically similar, which, if they are, would show that there's a process of contamination going through the plant. Or, for example, the same approach could be used on raw materials, that are coming into the plant, or inputs for growing fresh produce. So the first question really is, what are you using this for? For the second question, what are the technical, procedural, or equipment requirements? So this is, even though whole genome sequencing has become relatively mainstream, it is still quite technical and challenging. So if a member of the CPMA is thinking of incorporating this, there's several things to consider. The first is there needs to be sufficient lab capacity and sufficient space to do this. There has to be a dedicated lab, because contamination with other DNA fragments will really lead to complexities with DNA sequencing. And we know that DNA is in all living organisms, including plant material. So there has to be a dedicated laboratory where contamination issues will not happen. Then we have to talk about the reagents to do this. They often have a short shelf life. They require being frozen, and they require timely usage. Third is the cost. So estimates for establishing sequencing capabilities for WGS, or whole genome sequencing, vary from between $100,000 US dollars to up to $700,000 US dollars, depending on what's being done, the throughput of the isolates, and so on and so forth. The cost to sequence a single bacterial isolate, once a lab is set up, varies from between $150 to $200. And again, it depends on how many isolates are being sequenced at the same time. The more isolates are being sequenced, the cheaper the cost. The cost to set up the computational requirements are similarly varied, and also depend on the needs of the specific laboratory. So you can see that there's a lot of variables here, and it is quite technical. Next, we have to consider personnel. It's a technical procedure, so you have to have someone that's highly trained to do this. And that also includes personnel on the bioinformatics side. So you will recall that we talked about, once the DNA has been sequenced in these DNA reads, it has to be put together back into the whole genome like a puzzle. And then that genome has to be analyzed, for example, to see if two genomes from different environmental sources are the same. That requires trained people, appropriate software. Another consideration is the fact that, as I've said, this is enormous data that's being generated, so you have to have enough hard drive space to store this, which is not trivial. And so because of all this, I recommend that unless a CPMA member would be doing sequencing routinely, it's actually better to form collaborations with universities who do this, or to work with commercial labs who have sequencing setups.
I certainly appreciate the complexity of the process, and certainly the need for the specialized equipment. And to your point, certainly you want to make sure that whomever is running those procedures has the technical skills and the understanding to really protect the integrity of the data that is created. I have two more questions for you, and we'll try to get through these fairly quickly if we can. Dr. Farber, if I can just come back to you to begin with. Historically, we've used the epidemiological approach with respect to foodborne outbreaks and foodborne illness investigations. Do you see whole genome sequencing replacing the need for that? Or is it more of something that enhances the epi groundwork that needs to be done?
Yeah, thanks, Jeff. So I don't think that has really changed, Jeff. I think the epi investigations are still really critical to the overall analysis when you're doing a foodborne outbreak investigation. You may have seen Health Canada developed actually a weight of evidence document, factors to consider for appropriate and timely action in a foodborne illness outbreak investigation. This was done after the maple leaf listeriosis outbreak, but it's still really extremely relevant today. So you have not only your lab investigation, which gives you, your whole genome sequence of your strains, and not only the epi investigation, which is critical, because if you have, let's say, a case and five patients and they never ate the implicated product, that tells you that something is off in terms. But it's not only the EPI investigation, but it's the food safety investigation, which for large part CFMA does, as you're familiar with, both the trace back and the trace forward. So, for example, if the cases in Quebec that are being connected and the implicated product never went to Quebec, obviously something is obviously amiss there. So it's really the lab investigation, the food safety investigation, and the EPI investigation, which are all really critical and important in terms of the overall process of foodborne illness investigations.
Thank you for that. And I know in speaking with folks at the Public Health Agency of Canada and CFIA, I know they certainly appreciate the power of the whole genome sequencing. But you're right, it still comes back to doing those food histories and boots on the ground and pulling samples to find out the real root cause or real problem of where an issue has arisen. My last question, and I always like to put my guests on the spot this way, and this goes to both of you, so please feel free to jump in. As experts on the cutting edge of food safety, both policy, practices, and technology, can you kind of look into your crystal balls and give our members some insights into what changes they can expect over the next, let's say, 5 years when it comes to the whole genome sequencing and how you see it evolving?
So basically, the way I see it evolving, Jeff, is that it's going to get to the point where it becomes routinely used by food companies to solve many different problems. It's going to get much faster. I know Dr. Goodridge is using some of the latest techniques with the MinION, which very quick to get you a sequence of a particular strain. So I think the food industry is going to, for example, they'll be able to better understand why their food is spoiling, because it's not only the whole genome sequence, but if you're going to be sequencing, for example, as Dr. Goodridge mentioned, some of the bacteria in your plant. So you may want to look at what is the microbial composition of food in your plant. And if you have 2 or 3 different plants and one of your plants, you have a cheese that is spoiling for really unknown reasons that baffling people. If you do a what referred to as a microbiome analysis, so a full microbial analysis of that initial cheese and final cheese product, and you find out that the bacteria in that product that's consistently spoiling is very different from your other 2 plants, which you have no problems. I think this is going to be, very standard in terms of trying to help companies avoid some of these food spoilage problems. I think that in terms of whole genome sequencing by public health agencies, it's going to even get better because, I've had some discussions with some public health folks. They're going to be able to use, for example, both machine learning and AI to look at, for example, the whole genome sequence of a bacteria. And maybe that bacteria has caused 1 or 2 cases, but now they're going to be able to use machine learning or AI to predict, is this strain likely to cause further illnesses in the future or other issues which we never even suspected? So there are many avenues where I think sequencing in general, whole genome, next generation sequencing is going to make our lives easier. It's going to drive continuous improvement in food safety. But I think the food companies are going to have mechanisms in place to be able to take decisions based on the outputs from all the big data that is going to be come to us now and will be even more so in the future.
Thank you very much for that, Dr. Farber. And Dr. Goodridge, we'll give the last word to you.
Yes. So following up on what Dr. Farber has said, I agree that things are going to become increasingly streamlined and more mainstream, and methods will be simplified and so forth to make it easier for food companies to use, or fresh produce growers and so forth to use. And so the one area that I want to focus on is a method called metagenomic next generation sequencing. So we've really focused on whole genome sequencing in this talk, which, as I said, refers to taking a pure bacterial colony or bacteria that come from a pure bacterial colony and sequencing that. But we're now starting to see that mixtures of microorganisms can be sequenced at the same time. And so Dr. Farber touched on this when he talked about the microbiome, which has really become a buzzword. And we now, understand the importance of looking at microorganisms as they relate to other microorganisms in the same environment because they interact. And so, newer technologies, Dr. Farber touched on a sequencer called the MinION. This is actually a very small DNA sequencer you can put in your hand. And what it does is it actually delivers sequences in real time. And the company that makes this, their goal is really to get sequencers out into the hands of everybody. So they're really working to simplify approaches. And so in the context of what we're talking about today, I can envision the separation between rapid detection of pathogens using currently available rapid methods like PCR, for example, and sequencing being blurred. Because with this technology, you can actually do sequencing in real time. So it would be possible to actually use sequencing as a rapid detection method. But also attached to this is the fact that instead of just looking for one pathogen at a time, one could look for any pathogen that's present in the food at the same time. And this is important because we know, that if, for example, a fresh produce commodity is contaminated with feces from a wild animal, that there may be more than one pathogen that's present. And so this will really lead to an increase in food safety, in my estimation, and safer food produced.
Well, thank you. And then I do appreciate also you giving me an idea for a future podcast on metagenomic sequences. So I will make a note about that as well. Well, that comes to the end of the technological piece of this podcast. So on behalf of our CPMA members, I would like to thank Drs. Goodridge and Farber for your insights into this exciting new technology. Thank you very much and take care.
Thank you.
Thank you.
Now that we have a better understanding of the history, technical, and procedural nature of whole genome sequencing, I'd like to turn the discussion to the practical applications for our industry. To help us with this, I'd like to welcome Afreen Malik to the discussion. Afreen Malik is the Science Programs Director at Western Growers, where she is responsible for designing, developing, and deploying programs in support of key Western Growers science initiatives in food safety, environmental compliance, and data science. Prior to joining Western Growers, Afreen served as Director of Technical Services at International Food Safety and Quality Services, a consulting firm that helped companies produce safe foods. Before consulting, Afreen spent 20 years in technical services and quality assurance roles at Ocean Mist Farms, Sunrise Growers, and Calveno Foods. Afreen has also served as a biological assistant for the California Department of Food and Agriculture. Afreen has a Master's of Science in Coastal and Watershed Science and Policy from California State University, Monterey Bay, and a Bachelor of Science in Biological Sciences from the University of California, Santa Barbara. Welcome, and thank you for joining us, Afreen.
Good morning. It's great to be with you.
Great. So, as I noted, we've kind of talked about the technical side of whole genome sequencing. So now I'd really like to get your input with respect to how whole genome sequencing is changing our industry and kind of what you see for the future. So, whole genome sequencing has emerged as a powerful tool in recent foodborne outbreak investigations, but has also been used to uncover links to past outbreaks, such as the Bluebell ice cream issue. Is there anything which you find surprising or unexpected as this new technology becomes more widespread?
It's funny that you should mention how WGS can link an analysis you might conduct today to a historic outbreak. This is one of the things early on that, from my perspective, really surprised me and was unexpected, primarily based on my elementary knowledge of bacterial replication and the assumptions around the rate of mutation. That the bacteria 10 or 15 years from now could match something that we find in an environment today, right? So that was actually a surprise to me, because I expected bacteria to change over that time frame. So initially that was a surprise and something that I didn't expect, but that speaks to the power of this tool to be able to pinpoint this sort of a link. The other thing that I find interesting is that when you dig a little bit deeper into whole genome sequencing and what it can and can't tell us, and speak with some subject matter experts, you learn how the interpretation of the whole genome sequencing can vary between experts. So you quickly learn how important the metadata surrounding the sample is and how this can guide your interpretation of the results. So as you mentioned, whole genome sequencing is a powerful tool that can help us detect contamination with much more precision than we did in the past. However, I think it requires a certain level of understanding, not just about genomics and phylogenetics, but about the complexities within the fresh produce industry and the production environments. So I think those are kind of the two things that, sort of surprised me, not so much surprised me, but, are unexpected, that, that we have this precise tool that can give us, a great level of granularity in what we're looking at. But at the same time, there is this, somewhat of an ambiguity about how to interpret the results of that analysis. So I find that somewhat ironic as well. But I think those would be my two top unexpected consequences of this sort of a technology.
Yeah, it's interesting. It's so precise. And I sometimes, compare it to a scalpel. A scalpel is a very, very precise instrument, but it doesn't work very well if you want to take down a redwood. So a redwood tree. So, you have to know that each tool has its place and has to be used properly to really be a benefit to anyone. Like CPMA, Western Growers is an industry association. And I'm wondering what types of questions or concerns your members have expressed regarding whole genome sequencing and its impacts on the industry. And I know this is important because you obviously have a whole lot of the leafy green industry that falls under Western Growers. So I'm just kind of interested to know what your members are saying.
Yeah, no, that's a great question. So we get various questions related to whole genome sequencing. One of the key questions and concerns that come our way from our members are regarding opportunities to learn more about whole genome sequencing and what it means when the regulatory agencies identify a close match from a clinical sample to a particular food vehicle. What is a close match? What questions should I be asking if I'm in a similar situation? Should I use this tool in my own operations, and what are the risks, benefits of using it? So those types of questions come our way quite often. But I think the second most common concern that we hear about is linked to how one might expose their operation to regulatory or legal consequences if they use this tool. So just like with any other type of testing, an operation must understand why they are applying a particular technique or testing tool for a particular organism. Without clearly understanding this and the potential consequences of the testing, it would be irresponsible to move forward, in my opinion. So I think, this idea behind really understanding what you're testing for and what information you think you'll gain through that testing is a key concern and question that comes our way. And then, of course, with whole genome sequencing, that the legal and regulatory consequences are quite important. But a more practical concern has to do with the cost and the time it takes to obtain results from this sort of a tool, and to find local labs where this sort of testing could be conducted. So these aspects of whole genome sequencing are changing fast as the technology is scaling up, and I suspect the technology will continue to improve and costs will continue to decline over time.
All right, well, thanks for that. just want to build on a little bit. You've brought in, the what individual companies should be doing or thinking about. So if I can just kind of expand on that a little bit, what role do you see the whole genome sequencing playing from the 30,000 foot level, from the overall fresh produce food supply chain? And second to that is, do you see a routine role for fresh fruit and vegetable industry organizations to incorporate whole genome sequencing within their food safety operations? So if you can just kind of give us your thoughts from the big overall fresh produce supply chain system, 30,000 foot level, and then, how you see that playing out on a more granular individual organization standpoint.
Yeah. So, speaking with various members and just gauging my understanding of where the industry is in terms of how they're currently using whole genome sequencing and what their level of understanding is about this technology. I think as it stands now, the primary application of this tool would be in something like root cause analysis or other investigative purposes. I don't think that whole genome sequencing is going to be used for routine testing anytime soon. And that's not a bad thing because whole genome sequencing is not appropriate for all types of situations, right? You wouldn't want to necessarily employ whole genome sequencing for routine testing for various reasons, both related to cost, the time it takes to get the results, but also the type of information that you get. You don't necessarily need to know that level of granularity if you're trying to simply verify your existing protocols. So if you're trying to see if your sanitation protocols are effective, you don't need to have whole genome sequencing conducted on your samples. So, I like I said, I don't think that whole genome sequencing is going to be used for routine testing anytime soon. The objectives one has in routine testing programs, which are often related to verification, are not best met using this tool. There are other simpler and more familiar and cost-effective tools that work very well for routine programs.
Okay, thanks for that. And I think it's very interesting. As new technologies come along, some people kind of feel that urge to jump into it, and right, without necessarily understanding, what they're getting into. I'm just wondering if there are any particular watchouts that you would suggest that people be aware of in the event that they do want to incorporate whole genome sequencing into more of what they do.
Yeah, I think that's a very good question. My primary advice would be to first understand what you're choosing, why you're choosing this tool over others. What information will whole genome sequencing give you that other tools might not? And what will you do with this information? So in short, just understand what the consequences of whole genome sequencing might be under the current regulatory and legal environment, and what the pros and cons are for your organization. I would recommend that you speak to a subject matter expert and your legal team before you begin to implement whole genome sequencing in your program. I think that this is a very powerful tool, and it certainly has applications in the fresh produce industry, but you should understand what its limitations are and what its strengths are before you actually apply it in your operations.
So just kind of building on your comments around regulatory, Canada here, we have the Public Health Agency of Canada and the Canadian Food Inspection Agency. Of course, down south in the US, you guys have the Food and Drug Administration, as well as the Center for Disease Control. And we know those organizations have really taken to whole genome sequencing, and they recognize what a powerful, to use your word, technology it is, and they've built it into their investigative toolboxes. Is there anything which Western growers members or CPMA members should be prepared for if presented with whole genome sequencing data from those regulatory bodies, in relation to a recall or in relation to any other thing that it looks like the regulators are investigating you on?
Yeah, I was, I worked for a grower, shipper, processor, so I can relate to that question, because you're always thinking about, what would I do if I'm in this situation? What sort of questions should I be asking? What sort of resources should I be tapping into? The most important thing that I would advise, to our members is to remember that under this scenario, help is available to you. Reach out to your trusted trade organizations or associations to learn about and include contact information for subject matter experts on whole genome sequencing. Include this information in your crisis management plan so that your team knows exactly who to contact to help you interpret the information that's being presented by the regulatory agencies and to help you ask the right questions. So don't think that you're on your own. Whole genome sequencing is not something that everybody understands or that all operations have the expertise to react to in-house. So make sure that you have those connections outside of your organization and that you know who to contact if you are presented with this sort of a scenario. I would also encourage operations to educate their staff on the strengths and limitations of whole genome sequencing. This will help your operation be in a better place if you are in this sort of a scenario, and to be able to better handle regulatory inquiries that may come your way. So, I'm just looking at it from that perspective. I think that those are two key ways that operations can prepare to handle this sort of a request from their regulatory body.
Yeah, and your advice to include, the contact information for those experts that you can reach out to, and having that within your crisis management program before you are in a crisis is key, because we've all been in those situations where everything's flying at us left and right and very fast. So being able to open up a page in a binder and say, okay, here's who we need to talk to, really helps to kind of control the messaging, control the dialogue, and helps to get you to a spot where you can start to answer those questions from the regulators. So that certainly is excellent advice there. So the last thing I like to do to all my guests is kind of put them on the spot. And I'm going to ask you to look into your crystal ball and tell us what you think the future of whole genome sequencing is, and specifically, how do you see it influencing the fresh produce industry in the years to come?
I wish I had a crystal ball. I suspect that next generation sequencing will continue to evolve and improve. It may help us answer some important questions and fill some key knowledge gaps relative to produce safety. I think it's already helping us better understand how pathogens survive, move, and persist within growing environments, and what factors contribute to the movement of pathogens from the environment into our products. So I think as the technology scales up and becomes more widely accessible, I would expect that the cost would also continue to decline, as well as the time between sample collection and results would also decline, which would make it more practical and useful for the produce industry. So, with this and with a better understanding of what this tool can and can't tell us, I would think that there would be a wider adoption of this technology in the fresh produce industry, not just for the special case root cause analysis or investigative purposes, but maybe in other aspects of ensuring produce safety as well. As things scale up, as the technology improves in those various aspects related to time to results and costs, And, as I would imagine, the regulatory and legal environments might change over time. That probably will take longer than the technology. Yeah, it usually does. But I think all of those things, over time might help a broader adoption of whole genome sequencing in the produce industry.
Well, Afreen, I'd like to thank you very much for sharing your time and your insight and your knowledge with CPMA's memberships. It certainly sounds like the next few years will be very interesting, and certainly something for us to keep an eye on and follow as we move forward here.
Absolutely, I agree. Thank you so much for this opportunity. It's a pleasure being here with you.
Well, thank you. So once again, I would like to thank Dr. Goodridge, Dr. Farber, and Afreen Malik for sharing their knowledge, time, and foresight on the ever-evolving world of whole genome sequencing. So Wally, back over to you.
Thank you to Jeff and to our panelists for that informative podcast. Until next time, fill half your plate with fruits and veggies, continue to seek out new knowledge, and basically never stop growing.